Snails can have shells that spiral either to the right, known as dextral, or to the left, known as sinistral. In most species, dextral snails are far more common, while sinistral snails are rare. But why does this happen? The direction of coiling isn't random, it's determined by genetics, specifically through a phenomenon called maternal effect inheritance. The key gene responsible for this is inherited from the mother, but its effect is seen in the next generation, not the one carrying it. We'll break this down in the next slides. Before we dive into the genetics behind sinistral and dextral snails, there's an interesting case of a rare left-coiling snail named Jeremy. His unique condition led to a global search for a mate, but that's a story for another video. I've already covered it separately if you'd like to check it out. In the snail Limnaea peregrina, the direction of shell coiling is determined by a single gene with two alleles, capital D for dextral and small d for sinistral. But here's the twist. It's not the offspring's own genotype that decides their coiling direction. Instead, it's the mother's genotype that matters. If a snail's mother has the genotype small d, all her offspring will have sinistral shells, even if they inherit the dominant capital D allele. But if the mother is capital D, her offspring will have dextral shells, regardless of their own genotype. So, even though dextral is dominant over sinistral, the inheritance pattern follows a maternal effect, meaning the mother's genes control the trait of her offspring. When a dextral female is crossed with a sinistral male, all the F1 offspring have dextral coiling, suggesting that dextral is dominant over sinistral. Interestingly, the reciprocal cross, where a dextral male is crossed with a sinistral female, produces an entirely different result. Here, all F1 offspring coil to the left, becoming sinistral. The difference in F1 results, depending on the maternal parent, proves that shell coiling follows the rule of maternal or cytoplasmic effect inheritance. In the F2 generation, all females, regardless of their parental cross, are right-coilers. However, their genetic makeup plays a crucial role in determining the shell coiling of future generations. When F2 males are mated with different females, distinct patterns emerge. If an F2 male is crossed with a homozygous dextral female, all their offspring are right-coilers with no segregation. But when the same male is mated with a homozygous sinistral female, only left-coiling offspring are produced. Now, when these F2 females are crossed with males of any genotype, their F3 offspring finally reveal the expected 3-to-1 ratio of right-coilers to left-coilers. This delay in segregation further proves that a snail's coiling pattern is determined by the genotype of its mother, not its own genes. A snail shell coiling direction isn't determined by its own genes, but by its mother's genotype, a phenomenon known as the maternal effect. During egg formation, maternal nurse cells provide mRNA and proteins that persist through early embryonic development. If the mother carries at least one dominant D allele, these gene products guide the early cell divisions, resulting in dextral offspring. However, if the mother is recessive, only the small d gene products are transferred, leading to sinistral offspring, regardless of the embryo's own genotype. This early influence occurs because the maternal gene products control the orientation of the mitotic spindle during the first few cell divisions, determining the snail's final coiling direction before it even begins expressing its own genes. This phenomenon isn't limited to snails. Maternal effect genes play a crucial role in early development. In fruit flies, they determine body axis formation before the embryo's genes even activate. This phenomenon extends to mammals, including humans, influencing early embryogenesis. Want to learn more about fascinating biology concepts? Subscribe now.